Welcome to the fascinating world of genetics. Today we'll explore how DNA, RNA, and proteins work together to create the building blocks of life. From the tiniest bacteria to the largest mammals, every living thing relies on these molecules to function and grow. Understanding how genetic information is stored, transferred, and used is key to unlocking the mysteries of biology. Let's begin our journey by looking at the central dogma of molecular biology, which describes the flow of genetic information within a cell. This process is essential for life as we know it, and it all starts with DNA. DNA, or deoxyribonucleic acid, is the hereditary material found in almost all living organisms. It carries the instructions needed for an organism to develop, survive, and reproduce. DNA is made up of two long strands that coil around each other to form a double helix. Each strand is composed of smaller units called nucleotides, which include a sugar, a phosphate group, and a nitrogenous base. The sequence of these bases encodes genetic information, much like letters in a book. This information is passed from parents to offspring, ensuring the continuity of life. Double helix structure of DNA was famously discovered by James Watson and Francis Crick in 1953. This twisted ladder shape is stabilized by hydrogen bonds between complementary bases, adenine pairs with thymine, and cytosine pairs with guanine. The unique pairing ensures that DNA can be accurately copied during cell division. The double helix not only protects the genetic code, but also allows it to be efficiently packed inside the cell nucleus. This elegant structure is a marvel of nature and a cornerstone of molecular biology. Within the DNA molecule, specific segments called genes contain the instructions for making proteins. Genes are organized along structures known as chromosomes. In humans, there are 23 pairs of chromosomes in each cell. Each chromosome is a long DNA molecule wrapped around proteins called histones, which help package the DNA tightly. Genes vary in length and can code for different traits, from eye color to blood type. The arrangement and expression of these genes determine the unique characteristics of every individual. The process of turning genetic information into functional products begins with transcription. During transcription, a specific segment of DNA is copied into RNA, or ribonucleic acid. RNA acts as a messenger, carrying the genetic code from the DNA in the nucleus to the ribosomes where proteins are made. This step is crucial because DNA cannot leave the nucleus, but RNA can travel to other parts of the cell. Transcription ensures that the genetic instructions are delivered to the right place at the right time. RNA, or ribonucleic acid, is a molecule similar to DNA but with some important differences. Unlike DNA's double helix, RNA is usually single-stranded. It contains the sugar ribose instead of deoxyribose and uses the base uracil in place of thymine. RNA comes in several forms, each with a unique role in protein synthesis. The most common types are messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Together, these molecules help decode the genetic instructions and build proteins. There are three main types of RNA involved in protein synthesis. Messenger RNA, mRNA, carries the genetic code from DNA to the ribosome. Transfer RNA, tRNA, brings the correct amino acids to the ribosome during protein assembly. Ribosomal RNA, rRNA, forms part of the ribosome itself, helping to catalyze the chemical reactions that link amino acids together. Each type of RNA plays a specific and vital role in translating genetic information into functional proteins. Transcription is the first step in protein synthesis. It begins when an enzyme called RNA polymerase binds to a specific region of DNA known as the promoter. The enzyme unwinds the DNA and uses one strand as a template to build a complementary RNA strand. As the RNA strand grows, it detaches from the DNA, and the DNA helix reforms. The result is a single-stranded mRNA molecule that carries the genetic instructions needed to make a protein. For mRNA can be used to make proteins, it must be processed. In eukaryotic cells, this involves adding a protective cap to one end and a poly-A tail to the other. Introns, or non-coding regions, are removed, and the remaining exons are spliced together. This mature mRNA is now ready to leave the nucleus and travel to the ribosome, where it will guide protein synthesis. These modifications help stabilize the mRNA and ensure accurate translation. Once the mature mRNA reaches the ribosome, the process of translation begins. Translation is the step where the genetic code carried by mRNA is used to assemble amino acids into a specific protein. The ribosome reads the mRNA sequence 3 bases at a time, with each triplet called a codon. Each codon specifies a particular amino acid, and the sequence of codons determines the order of amino acids in the protein. This process is essential for turning genetic information into functional molecules. Genetic code is a set of rules that determines how the sequence of bases in mRNA is translated into amino acids. Each codon, made up of 3 RNA bases, corresponds to one of 20 amino acids or a stop signal. The code is nearly universal, meaning it is shared by almost all living organisms. This universality highlights the common ancestry of life on Earth and allows scientists to study genes across different species. Each codon in the mRNA sequence specifies a particular amino acid. For example, the codon AUG codes for methionine, which is also the start signal for protein synthesis. Other codons signal the addition of different amino acids, while 3 codons act as stop signals, ending the process. The precise order of codons ensures that proteins are built with the correct sequence of amino acids, which is critical for their function. Transfer RNA, plays a crucial role in translation. Each tRNA molecule has an anticodon that pairs with a specific codon on the mRNA. At the other end, tRNA carries the corresponding amino acid. As the ribosome moves along the mRNA, tRNAs bring the correct amino acids in sequence, allowing the ribosome to link them together and form a growing protein chain. This precise matching ensures accurate protein synthesis. Ribosomes are complex molecular machines made of RNA and proteins. They have two main subunits that come together during translation. The ribosome reads the mRNA, matches tRNAs to codons, and catalyzes the formation of peptide bonds between amino acids. This assembly line process continues until the ribosome reaches a stop codon, signaling the end of protein synthesis. Ribosomes are found in all living cells and are essential for life. Process of translation begins with initiation. The small ribosomal subunit binds to the mRNA near the start codon. A special initiator tRNA carrying methionine pairs with the start codon, and the large ribosomal subunit joins to form a complete ribosome. This assembly marks the beginning of protein synthesis, setting the stage for the addition of amino acids according to the mRNA sequence. During elongation, the ribosome moves along the mRNA, reading each codon and bringing in the corresponding tRNA. Each new amino acid is added to the growing protein chain through peptide bonds. This process continues, with the ribosome shifting and tRNAs entering and exiting, until the entire mRNA sequence has been translated. Elongation is a highly coordinated and efficient process, ensuring that proteins are built quickly and accurately. Translation ends when the ribosome encounters a stop codon on the mRNA. No tRNA matches these stop codons, so release factors bind to the ribosome instead. This triggers the release of the newly formed protein and the disassembly of the ribosome. The completed protein can now fold into its functional shape and begin its work in the cell. Termination ensures that proteins are made to the correct length and composition. After synthesis, proteins must fold into specific three-dimensional shapes to function properly. This folding is guided by the sequence of amino acids and sometimes assisted by helper proteins called chaperones. Proper folding is critical because a protein's shape determines its role in the cell. Misfolded proteins can lead to diseases, highlighting the importance of this final step in protein synthesis. Proteins are the workhorses of the cell, performing a vast array of functions. They act as enzymes, speeding up chemical reactions, and as structural components, giving cells their shape. Proteins also transport molecules, send signals, and defend against disease. The diversity of protein functions is made possible by the unique sequence and structure of each protein, all determined by the original DNA code. Sometimes changes occur in the DNA sequence, known as mutations. These can be caused by errors during DNA replication or by environmental factors like radiation. Some mutations have no effect, while others can alter protein function or cause disease. However, mutations are also a source of genetic variation, driving evolution and the diversity of life. Understanding mutations helps scientists study genetic diseases and develop new treatment. Before a cell divides, it must copy its DNA so that each new cell receives a complete set of genetic instructions. DNA replication is a highly accurate process involving several enzymes. The double helix unwinds, and each strand serves as a template for a new complementary strand. This ensures that genetic information is faithfully passed on to the next generation of cells. Several enzymes are involved in DNA replication. Helicase unwinds the double helix, while DNA polymerase adds new nucleotides to form the complementary strands. Other enzymes help proofread and repair errors, ensuring high fidelity. These molecular machines work together in a coordinated fashion to duplicate the cell's genetic material quickly and accurately. Central dogma of molecular biology describes the flow of genetic information. DNA is transcribed into RNA, which is then translated into protein. This sequence of events is fundamental to all living things. By understanding the central dogma, we gain insight into how genes control the structure and function of every cell in our bodies. Not all genes are active at all times. Cells control gene expression to ensure that proteins are made only when needed. This regulation can occur at many levels, from controlling transcription to modifying proteins after they are made. Gene expression control allows cells to respond to changes in their environment and carry out specialized functions. But genetics refers to changes in gene expression that do not involve changes to the underlying DNA sequence. Chemical modifications, such as adding methyl groups to DNA or altering histones, can turn genes on or off. These changes can be influenced by environmental factors and can sometimes be passed to future generations. Epigenetics adds another layer of complexity to how genes are regulated. Mutations can disrupt normal protein function and lead to disease. For example, a single base change in the gene for hemoglobin causes sickle cell anemia. Other mutations can increase the risk of cancer or inherited disorders. By studying these genetic changes, scientists can develop better diagnostic tools and treatments for a wide range of diseases. Genetic engineering is the process of directly modifying an organism's DNA. Scientists can add, remove, or alter genes to create new traits or correct genetic defects. This technology has revolutionized medicine, agriculture, and research. Examples include genetically modified crops, gene therapy for inherited diseases, and the production of insulin using bacteria. CRISPR is a powerful tool for editing genes with precision. It uses a guide RNA to target specific DNA sequences and an enzyme called Cas9 to cut the DNA at that spot. Scientists can then add or remove genetic material as needed. CRISPR has opened up new possibilities for treating genetic diseases and advancing biological research. Prokaryotic cells such as bacteria, DNA is not enclosed in a nucleus. Transcription and translation occur almost simultaneously in the cytoplasm. This allows for rapid protein production, which is essential for the fast growth and adaptation of bacteria. The basic steps of protein synthesis are similar to those in eukaryotes, but the process is more streamlined. Eukaryotic cells, like those in plants and animals, have a nucleus that separates DNA from the rest of the cell. Transcription occurs in the nucleus, and the mRNA must be processed and transported to the cytoplasm before translation can begin. This compartmentalization adds extra steps but allows for more regulation and complexity in gene expression. Alternative splicing is a process where a single gene can produce multiple proteins. By including or excluding different exons during mRNA processing, cells can generate a variety of proteins from the same DNA sequence. This increases the diversity of proteins and allows organisms to adapt to different conditions. After proteins are synthesized, they often undergo further modifications. These can include adding chemical groups, cutting the protein into smaller pieces, or attaching other molecules. Post-translational modifications can alter a protein's activity, location, or stability, allowing cells to fine-tune their functions. Proteins do not last forever. Cells have systems to break down and recycle old or damaged proteins. This process, called protein degradation, helps maintain cellular health and balance. Specialized structures called proteasomes recognize and destroy proteins that are no longer needed, ensuring that the cell's machinery runs smoothly. Proteomics is the large-scale study of proteins, their structures, and functions. By analyzing the entire set of proteins in a cell or organism, scientists can gain insights into how genes are expressed and how cells respond to different conditions. Proteomics is a rapidly growing field with applications in medicine, agriculture, and biotechnology. Genomics is the study of the entire genome, or all the DNA, of an organism. Advances in sequencing technology have made it possible to read and analyze genetic information on a massive scale. Genomics helps scientists understand how genes interact, how traits are inherited, and how genetic variation contributes to health and disease. DNA sequencing is the process of determining the exact order of bases in a DNA molecule. This technology has revolutionized biology, allowing scientists to identify genes, study mutations, and trace evolutionary relationships. Sequencing is used in medicine, forensics, and research, providing a powerful tool for understanding life at the molecular level. Human Genome Project was an international effort to map all the genes in human DNA. Completed in 2003, it identified over 20,000 genes and provided a blueprint for understanding human biology. The project has led to breakthroughs in medicine, genetics, and our understanding of evolution. It continues to inspire new research and discoveries. Understanding DNA, RNA, and protein synthesis has transformed medicine. Genetic testing can identify inherited diseases, while gene therapy offers hope for treating genetic disorders. Personalized medicine uses genetic information to tailor treatments to individual patients. These advances are improving health outcomes and changing the way doctors approach disease. Technology uses knowledge of DNA, RNA, and proteins to develop new products and technologies. Examples include genetically modified crops, biofuels, and synthetic biology. These innovations are helping to solve global challenges in food, energy, and health. As our understanding of molecular biology grows, so do the possibilities for biotechnology. DNA analysis is a powerful tool in forensic science. By comparing DNA samples from crime scenes to those of suspects, investigators can identify individuals with high accuracy. DNA evidence has revolutionized criminal investigations, helping to solve cases and exonerate the innocent. The reliability of DNA testing makes it a cornerstone of modern forensics. Genetic information provides a record of evolutionary history. By comparing DNA sequences across species, scientists can trace relationships and understand how life has evolved over time. Mutations and genetic variation drive evolution, leading to the diversity of life we see today. Genetics is a key tool for studying the origins and development of species. Advances in genetics and biotechnology raise important ethical questions. Issues such as genetic privacy, designer bodies, and gene editing require careful consideration. Scientists, policymakers, and the public must work together to ensure that new technologies are used responsibly and for the benefit of all. Future of genetics is full of promise. New technologies are making it easier to read, edit, and understand genetic information. As research continues, we can expect breakthroughs in medicine, agriculture, and beyond. The study of DNA, RNA, and protein synthesis will remain at the heart of these advances, shaping the future of science and society. Let's review what we've learned. DNA stores genetic information, which is transcribed into RNA and then translated into proteins. These processes are fundamental to life, driving everything from cell function to evolution. Advances in genetics and biotechnology are transforming our world, offering new solutions and raising new questions. By understanding the basics of DNA, RNA, and protein synthesis, we gain a deeper appreciation for the complexity and beauty of life. Thank you for joining this exploration of DNA, RNA, and protein synthesis. The journey from genetic code to functional protein is one of nature's most remarkable processes. As you continue your studies, remember that every cell in your body is carrying out these steps right now. Stay curious, keep learning, and discover how the secrets of genetics shape the world around us.